I was very pleased to get this introduction and also to get this invitation to present at the user meeting. And it really got me thinking about how long I've been coming to the ESRF. So for me, it's been over 20 years now. And the ESRF has really kind of formed the hub of a lot of my research. So as Guillermo mentioned, I started coming to the ESRF in 2001 when I was still a thesis student out in California at the Salk Institute. And I was introduced to the ESRF by Jean-Luc Ferrer, who unfortunately passed away about a little over a year and a half ago. But he was a visiting scientist in the lab. And so he took some of the thesis students from the lab to France, to Grenoble, where we had the opportunity to collect data here. And ever since, I've been coming back. So I came back as a postdoc and worked with Stephen Cusack at the EMBL. I worked at the ESRF for a while as a scientist. And since 2013, when I entered the CNRS, I've been right next door at the CEA campus. And so I regularly come to the ESRF. So I think this slide is pretty appropriate, with the ESRF kind of forming the center of a lot of my research for diverse projects over the past 20 years. But one thing I also want to really highlight is the fact that not only is the ESRF great in terms of the beamlines and the infrastructure, but also the scientists that work here. So I've had a number of very good collaborations currently with Mark Tully, who's a beamline scientist on the SACS beamline. A lot of the atomic force microscopy that we do is due to connections I made at the ESRF, in particular with Luca Costa, who was actually a thesis student when I met him with Fabio Komen, and we continue to work together. So it's been over 10 years now. And then of course, Max is now a long-term collaborator at the ESRF. So I'm not going to give much of an introduction about climate change, because we had that fabulous talk by Valerie just a few minutes ago. But one thing that we're interested in is how plants respond to changes in temperature. And so we do have these weather extremes. We also have this increase in temperature, which is quite significant for plants. So even a change of a degree or two can really change the way that a plant develops. So over here is a picture of Arabidopsis Thaliana. So that's our favorite little model plant that we use in the lab. And you can see that there's a big change in the growth and development of the plant, whether it's grown at 22 degrees on the left or 27 degrees on the right. Okay, so these plants are the same age. They were just grown at different temperatures. So this is a 5-degree temperature difference. So that's pretty significant, but you'll still see these differences even with 1 or 2 degrees. So you can see that you have something that's still in the vegetative phase, that's producing a lot of leaves, a lot of biomass, and then the plant grown at higher temperatures has quickly entered into that reproductive phase and has started to flower. So we're very interested in understanding what this mechanism is of how these plants know that this temperature is different and how that in turn changes their morphology. So these differences that we see in terms of changing their growth and development temperature, this happens very, very early on, so right after germination. So you can see here at the left, if you have an Arabidopsis plant grown at 20 or 28 degrees, you have this elongation of the hypocotyl, so that's the embryonic stem, and you also have this elongation of the petiole, so that's the stalk that attaches the leaf to the stem. And again, you see this all throughout development. So you have this elongation and you have this early entry into flowering. So this is true for our model plants, but it's also true for crop plants. So if we take a look at things like cabbage and broccoli, which are related to Arabidopsis, again, we see the same elongative growth under higher temperatures. And again, we see this for things like tomato as well. So this elongative growth that we see, a lot of this has been hardwired into plants during the course of evolution. And it's really a response to shade. So it's a shade avoidance response, this elongative growth, because the temperature is warm. So the plant thinks it should be getting some light, And one reason it wouldn't would be if it was shaded, for example, by competing plants around it. So it has this elongated growth to try to search out light in terms of enabling it to grow and reproduce. So this has effects on model plants. It also has important effects on our crop plants, this increase in temperature, because you have this fast entry into the reproductive phase. So you get early flowering and you get early seed and fruit production. But because there's less biomass, this can result in fewer and smaller seeds, again, less leaves, less biomass, and that can in turn translate to lower reproductive success. Okay, so there's important potential effects on crop productivity. And that's something that we need to both understand and potentially address. So why do plants have this response? How are these signals incorporated into plant development? Well, a lot of this goes through the circadian clock. So just like humans have a circadian clock, plants also have a circadian clock. So their metabolism, their leaf movements, that's all sort of tuned to day and night cycles. And so you have these repeating oscillations in gene expression, which occur over 24 hours. And this is really a big adaptive advantage for an organism because it can anticipate changes to day and night, as well as seasonal changes. So that's quite important. And actually about 30% of the genes in Arabidopsis are under circadian control. So they have this type of oscillative expression and repression over the course of 24 hours. So the circadian clock acts by having different genes which are expressed at different times, and they repress each other. So you have these oscillations which occur. So this is a very simplified schematic of the plant circadian clock, where you have certain genes which are expressed in the morning, and those repress genes which are expressed in the day and the early evening. And then you have genes expressed in the night, and you can see these lines here. These are all examples of genes which repress each other. So when you get these types of repression and expression patterns, these tend to oscillate over about 24 hours. Now, this clock can be tuned by environmental signals. And so big environmental signals are, of course, light during the day. So certainly during the summer, the day is longer and the night is shorter. So these light signals will actually tune gene expression. So we change that 24-hour oscillation of the clock a little bit. And then in the evening, a big signal is temperature. So those warm evenings in particular will really change gene expression of the circadian clock. And those temperature signals enter the circadian clock through this complex here called the evening complex. So this is a complex which we first started to focus on as a way to get these signals into the circadian clock and change downstream gene expression. We didn't know the mechanism of how this worked. So what's in the circadian, or what's in the evening complex? It's made up of 3 different proteins called loxarithmo, and that's a DNA binding protein. So that protein actually binds different regions of the genome and represses target genes. Okay, so this is the protein in the evening complex, which drives the entire complex to specific regions of the genome. And then it has 2 other proteins in this complex, one called early flowering 3 and the other called early flowering 4. So early flowering 4 is a small alpha helical protein, which seems to just kind of help glue everything together. And early flowering 3 is a big, long scaffold protein with no domains of known function and is plant specific. Okay. And these evening complex components are highly conserved plants. So even from mosses, you can find these proteins. So this is sort of an essential part of the circadian clock, and it's conserved over the course of evolution. So it's a quite important function in plants. So all 3 components of the circadian clock are expressed at about the same time, which makes sense because they need to work together in order to repress their target genes. And the question is, of course, how do they do that? So there are some indications that the evening complex is really important for physiological temperature sensing. We wanted to see if this was true. We wanted to study it in vitro so we could understand the mechanism of how this worked. So we needed to understand the role of the different proteins in the complex, as well as how the whole complex worked together. And the idea was, okay, maybe we can just discover this mechanism of temperature sensing. Maybe there's a little structural switch. We can use our regular tools of protein crystallography to understand the structure, for example. Sorry, my phone has started to ring. So the evening complex mutants have the same type of phenotype at 22 degrees. So if we take a look at a wild type plant, if we grow it at 22 degrees, it's shorter. And if we grow it at higher temperature, it has an elongated hypocotyl. Okay, so it's sensing the temperature change. So that's normal plant behavior, that's normal plant development. Now, if we take out one of those genes, so if we knock out the L4 gene, the L3 gene, or loxarithmo, we get the same phenotype. So the plant can no longer tell if it's low temperature at higher temperature. The evening complex is no longer able to form. It's no longer able to repress genes which are involved in growth and flowering. So you get this elongation of the hypocotyl even at lower temperatures. And if you grew these plants up, they would also flower very early. Okay, so this suggests that, okay, the evening complex has to work together as a complex. And each protein, if you get rid of any of them, the complex is no longer functional. Okay, so we know that if you get rid of the evening complex, plants can no longer sense those small changes in ambient temperature, or at least our model plant, Arabidopsis Thaliana. But we were curious, okay, is this a direct effect? Is this really the temperature sensor? And so how can we tell? So one thing which our collaborators did was to look at where these proteins bound in the genome. Okay, and to do this, they used an experiment called ChIP-seq, or chromatin immunoprecipitation, followed by sequencing. And so what you do for an experiment like this is you have your protein of interest, which binds DNA. So you basically take your plant tissue, for example, and you cross-link your proteins bound to DNA. You chop up your DNA, you shear the DNA into small pieces, and then you use an antibody specific to your protein of interest, and you pull out those protein DNA complexes, and then you sequence the DNA. So this gives you an idea of where a specific protein will bind in the genome. And so they did this for L3, L4, and loxarithmo. And they bound, as you can see here over on the left, they bound at the same places in the genome, which makes sense because we know they form a complex, and we know this complex actually has to have all these three proteins working together in order to have some effect on gene regulation. Okay, and then they did something else. They also tested this at plants grown at different temperatures. And what they saw, for example, for loxarithmo is that there are a lot more sites bound at 17 degrees than there were at 22 degrees. And they also did this for L3 and L4. So here, just take a look at the green curve and the blue curve. So this is the average binding intensity of all those peaks in the genome. So wherever L3, for example, is binding, in green is the binding at 22 degrees. And in blue is the binding at 27 degrees. Okay, so you have this big decrease in this binding intensity as the temperature increases. So this means this complex is no longer able to bind DNA, or it's binding DNA less well as we increase the temperature. Okay, and then here over on the right, again, is another experiment which shows the same thing. So plants were grown at 22 degrees, and then the temperature was shifted to either higher temperature or lower temperature. And that same experiment was run to see where those things were binding and if the binding intensity changed. And it does. So if you shift plants to lower temperature, your binding intensity increases. So you have this evening complex better bound at those lower temperatures. If you change the temperature to higher temperatures, you have the opposite effect where you have less binding to all your sites in the genome. So this really suggests that, okay, the evening complex is really implicated in this temperature response. Temperature changes the way it binds genome-wide. Now, is this something inherent to the complex itself, or is it some other ternary factor which is coming in to change its binding? That we don't know, but at least we have a very good indication that this is a major player in ambient temperature sensing in plants. So in order to see if this was a direct effect, we wanted to study the evening complex in vitro. So we reconstituted the entire complex. This was actually a lot of work by a postdoc in the lab, Katerina Silva, who spent a number of months working on reconstituting this complex because these proteins are not terribly soluble, various refolding protocols, but eventually she was able to reconstitute this complex. And so we were able to see what's called a band shift. So here we take a piece of DNA, which we know the evening complex binds, and that's down here, and we label it with a fluorophore, and we run it on a gel. And so that'll migrate fairly quickly on a gel. And then we add our proteins of interest, and if they bind, they'll slow down the migration of that DNA through the gel. And so here you can see this band up here corresponds to a shift where it's DNA plus the proteins of the evening complex. And so if we have, for example, ELF3 or ELF4 alone, they don't have DNA binding domains, so there's no shift in DNA. Lux in this type of gel actually has kind of a smeary diffuse band, but it does bind DNA. But really you have a nice stable complex when all three partners are present, and we can see that on a gel. So at least we can reconstitute in vitro a functional complex, which was a big advance in terms of in vitro studies of these proteins. So the big question was, okay, so we can reconstitute the complex, so can we also recapitulate that temperature sensitive DNA binding? And we can. So this is work by another postdoc in the lab, Stephanie Hutin. And what she did was she ran those same band shift assays, but at different temperatures, so 4 degrees, 15 degrees, and 27 degrees. And what she could show was that the complex bound quite well at lower temperatures, so at 4 degrees. There's a little bit of a shift at 15 degrees. It's kind of a fake band here. And then at 27 degrees, she didn't see any binding anymore. Okay, so it does seem like this complex is really important for temperature sensing, and it's a direct temperature sensor. So in vitro, we see that same behavior that we saw in vivo. So we have 3 possibilities. Maybe they're all working together as a temperature sensor, or maybe it's one of the proteins. So we're actually able to characterize the proteins separately as well using various structural and biophysical techniques. So Luxarithmo is the only one that directly binds DNA because it has the only DNA binding domain. And so we could test its DNA binding at different temperatures, and it didn't change. So the KDs were all the same over this ambient temperature range. So this one was not able to sense temperature, it just drives that complex to the DNA. L4 is a nice 100 amino acid alpha helical protein with a lovely alpha helical CD spectra. And again, that didn't change over this ambient temperature range. So that left us with ELF3. So for a protein crystallographer, a largely unstructured protein is not an ideal candidate for structural studies, but we decided to pursue it anyway. So we want to understand how this large scaffold protein was able to impart this temperature sensitivity, this temperature sensitive binding to the evening complex. So taking a look at the primary sequence of ELF3, so it's about 700 amino acids. There's very, very little secondary structure. There's a small alpha helix, and that's about it. It's mainly intrinsically disordered. So it has these large intrinsically disordered regions. Based on some prediction servers, it has a prion-like domain or prion-like region. And it's also predicted to phase separate. So that got us thinking, well, maybe liquid-liquid phase separation could be a mechanism for temperature sensing. And of course, this liquid-liquid phase separation, it's really kind of an in vogue concept right now. And it's been implicated in a lot of different physiological mechanisms. So it can be important for sequestering molecules. So it's a really good way to change activity. So we think that's important for function of the evening complex, that L3 can sequester its partners and they're no longer active. Liquid-liquid phase separation can also concentrate things. So you could have different enzymes, for example, concentrated into a liquid droplet, and that could improve enzymatic activity, for example. And liquid-liquid phase separation, it's also a general mechanism potentially to organize different cellular machinery. So there's a lot of functional roles which have been shown for phase separation, or potential functional roles. I would say that right now it's being used to answer perhaps too many biological questions, but we'll see. So phase separation for us was really, really attractive because it's a mechanism which is very, very sensitive to the physicochemical environment of the cell. So in vitro, if we look at proteins that phase separate, they'll do it as a function of protein concentration. They'll do it as a function of ionic strength. Changes in pH will change phase separation capabilities, and temperature does that as well. So phase separation was a very, very interesting potential mechanism, and our protein was predicted to undergo phase separation. So we want to characterize this in vitro as well as in vivo to see if this was a potential answer of how plants could sense temperature using these proteins of the evening complex. So taking a look at the sequence of ELF3, so about 300 amino acids of ELF3 is a prion-like protein. So it has some characteristics of yeast prion proteins. It has polyglutamine regions, and these polyglutamine regions actually vary with different ecotypes, different varieties of Arabidopsis. So they can be longer or shorter polyglutamine stretches, going from about 7 in our lab strain of Arabidopsis Thaliana to naturally occurring populations of Arabidopsis, which can have 29 polyglutamines. So they have a big variability, and they have a few little stretches of these polyglutamine regions embedded in this prion-like domain. And this was what was predicted to be important for phase separation. So we really focused on this region of the protein. So, we had our great hypothesis that ELF3, okay, it's probably the direct temperature sensor. And liquid-liquid phase separation would be a really nice mechanism for that. Can we study this in vitro? Can we tune phase separation based on our knowledge of the amino acid sequence and what are interacting and what's important for phase separation? And then if we can change these things in vitro, does this translate to changes in development in the plant? We can study things in vitro, but that may or may not be important in vivo, or may not occur in vivo. So we had some basic questions for both in vitro studies as well as in vivo studies. So first we needed to produce this protein and then see if it phase separated. And so we were able to do that. And so if you take a look on the right here, so this is ELF3 prion-like domain at 22 degrees and 27 degrees. So there's a bit of a change in terms of phase separation when we increase the temperature. And then if we take a look at 22 degrees, and if we take that prion-like domain and we mutate it, then we're actually taking a prion-like region from another species, which is not predicted to phase separate. So now we can have a version of ELF3, which is predicted not to phase separate, and indeed in vitro does not undergo phase separation. So we have a nice control. And so that doesn't phase separate under any of the conditions we tested where the wild type protein does phase separate. And so here you can see an example where we've taken ELF3 and we've fused green fluorescent protein to it. And then we can sort of track it using confocal microscopy, and we can see the formation of these little droplets, and we can see them come together and fuse. So it's really exhibiting liquid-like behavior, and it's certainly phase separating in vitro. And again, this is just another example of these droplets fusing, and this is kind of a classical experiment which people do to show that these biomolecular condensates do have liquid-like behavior and that they're fairly dynamic. So here's just an example of some still photos where you can see two droplets fusing. And then, of course, the other experiment, which is routinely done, is fluorescence recovery after photo bleaching. So again, we take our protein, which phase separates, so our different ELF3 constructs, and we fuse green fluorescent protein to it. And then using confocal microscopy, we can visualize those samples, and then we can use a laser to bleach either a portion of the droplet or the entire droplet, and then we can look at the recovery over time, and we can see if these drops recover over time. So you can see here, this is a small region which was bleached, and then you have sort of dynamic rearrangement, and you get this recovery. And so recovery can come from within the droplet, where you have sort of these molecules reorganizing, or you can have transfer from the outside. So you can have those two things, and those are two ways to recover. And this recovery happens, in this case, over seconds to minutes. And so here again, there's an example of some fusion, and here's our FRAP experiment. And so we're interested really, in environmental sensing and temperature sensing, and the effect of changes, particularly in this poly-Q region. So what we did is we made some different mutants in that poly-Q region where we lengthened or we shortened the poly-Qs, and we took a look at how these new constructs behaved as a function of both pH, protein concentration, and temperature. And so down here, depending on the mutant we use, we have a broader pH range, for example, where the protein would phase separate. And if we take a look at temperature, which is what we're really interested in, depending on how long that poly-Q region was, or if we just remove the poly-Q region, we'd either have behavior where the protein behaved like a switch, where it would go from diffuse to a condensed phase really quickly with a very, very sharp change at a very specific temperature, or then our wild type protein, which is in green here, had a more gradual change. And so as we changed that poly-Q region, we would have a very different phase separation behavior. And then down here, you can see an example where we're changing the temperature, and you can see the formation and follow the formation of liquid droplets. And this is reversible as well. So you have phase separation. As you increase the temperature, you're not changing anything else. If you change different variables, like pH, for example, or salt concentration, again, you can trigger the same phenomenon, the same phase separation of the protein. So of course, one question, if this is an environmental sensing mechanism, it'd be nice if it's reversible, because that would be a quick way to really change the function of a protein. And so we studied this, and again, this is an example of the wild type ELF3 from our lab strain of Arabidopsis. Again, you have this nice curve where you increase the temperature and you get this phase transition. And you can change the temperature back down. So if you do these temperature ramps up and down, the protein goes in and out of this diffuse, this dilute phase and this condensed phase. So that was nice. And then as a control, if we basically removed that prion-like domain, we changed that sequence with a different species, which doesn't have a prion-like domain. Again, there's no phase separation, so that's just our control there. Okay, so we've established in vitro that ELF3 does phase separate, it's reversible, and it's temperature dependent. But one thing that we did notice was that there was an aging process going on. So even when we had very circular or spherical shaped droplets, depending on how long they were there for before we tested them in a FRAP experiment. Depending on the pH or the conditions we used to prepare them, we would have slow to no recovery after photobleaching. So this suggests that even though you have something which looks like a droplet, it's no longer dynamic. So we're losing the movement of these molecules in the droplet. And so we're very curious as to what's going on. Is there something structural going on within these droplets where we're considering this liquid-liquid phase separation, but how dynamic is it and how much of a liquid is it staying? So as structural biologists, we wanted to structurally characterize this condensed phase, which is kind of difficult for something which, shouldn't be folded and should be dynamic. But we do have hints that it's not so dynamic. So how can we structurally characterize this? Well, I couldn't do protein crystallography, but we can do small angle X-ray scattering. So this is a collaboration with Mark Tully here at the ESRF, who was happy to help us try to understand what's happening in these phase-separated states. So if we take a look at ELF3, and these are different constructs with different length, polyglutamine repeats. If we take a look at these using small angle X-ray scattering, we get sort of a normal looking Sachs curve. I mean, they all look fairly similar to me. I will be honest, as perhaps a non-Sachs expert. And then if we look at the Kratky plot, we're kind of surprised to see that these things actually look pretty globular. So this is the dilute phase of ELF3, and we actually pass these over an FPLC column on the Sachs beamline. Okay, so we have stuff which we've removed aggregates. We have a fairly homogeneous sample, and this is before phase separation. So, so our proteins, which are predicted to be intrinsically disordered, which I would expect to be sort of an extended conformation, actually are relatively globular, and they seem to be small monomers. So that was, or small oligomers. So that was a surprise. And then it was a bigger surprise when we looked at the condensed phase. So what we saw when we took this condensed phase, so after we've undergone liquid phase separation and put it on the Sachs beamline, we saw the appearance of a peak in the Sachs curve. Okay, so the structure factor peak really suggests that there's some long range ordering of these molecules within this condensed phase. And perhaps even more interesting is that when we change the temperature for this experiment, so if we went from 4 degrees up to 22 degrees, this peak increased. And then if we went back down to 4 degrees, it would decrease. So you have this reversible phenomenon in the phase separated state, whereas you increase that temperature, you trigger phase separation, and you also seem to trigger ordering within this phase separated state. So that was a big surprise. And this was sort of one of the nice techniques here at the ESRF that we could use to study it. So I will say that some of this data was collected at Diamond Light Source during the shutdown at ESRF, but we'll be continuing these experiments here. So this is just a schematic to give you an overview of what we think is happening. So at lower temperatures in this dilute phase, these proteins are actually fairly compact. So kind of compact globular proteins, probably small oligomers based on the radius of gyration. And then as we increase the temperature and we trigger that phase separation, these molecules are adopting some type of ordered structure, and they're probably opening up and forming some kind of ordered stacking. And so we could use other techniques as well, a lot of them available here at the ESRF, to take a look at what's going on. So when we have these samples and we let them age for long enough, they actually go into a gel phase. And we can actually take a look at that gel phase again on the Sachs beamline. And that same structure factor peak that we saw in those solution samples, we see that same peak. It's just a bit more pronounced. Okay, now if we age these samples long enough, they're no longer reversible. So they don't actually go back into the dilute phase. They really form a pretty sticky, non-dynamic hydrogel. And it's no longer nice little round droplets. But we do have the same structure factor peak. So it does suggest, again, that even very early on, right after phase separation, this liquid is starting to really adopt a lot of order. And we can use electron microscopy actually on some of these hydrogels to really see or visualize that long range order. So these are different samples for our different constructs. And again, you can see that these molecules are really forming a highly ordered structure. And we could even see this in powder diffraction for a powder diffraction ring, which was collected on ID23 too. So here you can see this powder diffraction ring. And this is the kind of thing which you would see for prions, and you'd see it for other proteins which form kind of these extended beta structures. So we do see some of this very long range ordering, and we can see it at different resolution scales. So, when we think of a dynamic liquid-liquid phase separation, well, this aging process really does tune and change the physical properties of this phase separated state. So to go a little bit further into understanding what was happening, because a lot of times you'll see papers and you'll have these beautiful kind of spherical droplets, and they don't recover after photo bleaching, but they're still considered liquid-liquid phase separation, so maybe they really shouldn't be. So we want to kind of understand as much as we could about the properties of this phase separated state and the biomechanical properties. And to do that, we used atomic force microscopy. So this is a collaboration with Luca Costa in Montpellier, and he was for a number of years at the ESRF as a thesis student, postdoc, and maybe even a BMS or a scientist at the ESRF. But one thing that Luca does is he builds microscopes, and he builds very clever microscopes. So here he has an atomic force microscope and a confocal microscope. So we can look at the fluorescence of the sample, as well as measure the topography of the sample using AFM at the same time. So we know exactly what droplets we're looking at, and we can really get a lot of information on the sample this way. So this is an example here of some of the data that we collected for different constructs with different Q lengths, which we know kind of changes the properties of that phase separated state. So with AFM, we can see the topography of the sample. With confocal microscopy, we can look at the fluorescence because we're using these proteins with fluorescent tags. And then we can measure the stiffness of the sample. So we can measure some of those biomechanical properties of the same sample at the same time. And so if we do really kind of high resolution experiments, high resolution atomic force microscopy, and this is a turf microscope, so we can take a look at the fluorescence of the samples, and we can also measure their biomechanical properties of those same droplets. So these droplets still stay very spherical, but if we look at the biomechanical properties, they have different microenvironments. So they're very inhomogeneous within the sample. They have this step-like profile, suggesting that they have a nice layered structure, which is what is suggested by Sachs experiments, what we see in electron microscopy experiments. And they also have variable stiffness. So the biomechanical properties change with the length of the poly-Q. So longer and shorter poly-Qs change the stiffness and change those intermolecular interactions within the droplet. So just to kind of summarize, we've really tried to develop different tools which are available here at the ESRF to characterize droplets in this liquid-liquid phase separation, which goes from really a diffuse dilute phase to a dynamic phase for a while and then quickly ages into really a very, very stable hydrogel. But we can use different techniques to monitor that entire continuum of that process. And so, yeah, so we're really working on trying to describe this condensed state and to monitor it, which is something which is a bit tricky. So we have all this in vitro data. So does this mean anything in terms of plant physiology? So I mean, that's kind of the big question. I mean, this is interesting from a biomaterial standpoint, but what about in vivo? You know, do we have any evidence that phase separation is occurring and is important in terms of plant development, in terms of temperature perception? So our collaborators and us have done a number of different experiments to try to address that question. And it's a question we're still addressing, okay? I would say we have lots of preliminary data, which is very interesting. I wouldn't say it's proven yet, but I think we're going in the right direction. So we can use those same type of constructs where we tag our protein of interest, ELF3 in this case, with GFP, with green fluorescent protein, so we can track where that is in the cell. We can express it in different cells, so we can make yeast express our plant protein, and we can see what it does, and we can monitor its behavior. So this would be ELF3, which is a plant-specific protein without its partners. So this gives us a good idea if alone it can undergo phase separation in a cellular environment, which is very, very different from in vitro. Okay, so I think that's something that one shouldn't forget, that when we study things in vitro, it's very different than what's happening in a physiological context. And so here we can see that we have very, very nice formation of a condensate. So it's highly fluorescent, it's spherical looking. So we see the same type of behavior in vivo that we saw in our in vitro studies. So that was nice. And now what about in Arabidopsis, this actual plant host where this protein should be expressed? Well, we can take a look in Arabidopsis roots. And what we can see is that at lower temperature, we have a very diffuse fluorescence signal. So that stuff is present in the cell, but it's not forming a phase separated state. It's not forming a droplet. Now, if we increase the temperature of these plant roots, what we can see is we see the formation of these nice, very distinct speckles, suggesting again that we do have phase separation. And a very nice control which was done was to use a form of ELF3, that mutant form which I showed before, where we took away that prion-like domain and put a domain from a different species that's not considered a prion-like domain and not predicted to phase separate. And again, we don't see that phase separation. Okay, so in vivo, ELF3 is behaving the way we saw it behave in vitro, which is nice, gratifying, and very important to know, I would say. So ELF3 in vivo does form a phase separated state, and that formation is temperature dependent. And it depends on the sequence of that prion-like domain. So here, again, is just an example where if we grow these plants up and we change the sequence of that prion-like domain and make it no longer able to phase separate, the plant no longer is able to sense those differences in temperature. So if we grow Arabidopsis Thaliana at 27 degrees, this is what it should look like, okay? At this age, it should be flowering. But if we change ELF3, we no longer allow it to phase separate. So it seems to really kind of maintain this evening complex in a very active form. So this complex is repressing flowering. So even though it's quite warm, the plant no longer has that temperature perception. And that's just due to changes in this prion-like domain, in this polyQ region of ELF3. Okay, so just to kind of quickly summarize some of the work that we've done over the past 4 or 5 years now, ELF3 undergoes liquid-liquid phase separation, both in vitro and in vivo. And we're really working very hard to develop techniques to study this, particularly in vitro. And that required a lot of SAX experiments, atomic force microscopy. We're working on some NMR experiments, maybe PCS. These could all be very important techniques to really follow the dynamics of phase separation. The next big challenge which we have is, of course, in vivo. Because we study things in vitro, but it's a highly simplified system. And I think, as any protein crystallographer would note, just about any protein will phase separate. We see that all the time. And a lot of times it's not physiologically relevant. So I think we don't want to forget that. So we do need techniques to study this in vivo. And I think that's the next big challenge for us. And so based on our in vitro studies, we kind of know how this protein works. We're sort of starting to determine at the amino acid level what's important for phase separation. We can start to tune when phase separation occurs as a function of temperature. And so the question is, do we have a correlative physiological response? So can we change the temperature perception of our model plant Arabidopsis Thaliana as a function of what we've done to the PRD domain of L3? And so those are our experiments which are ongoing to really sort of correlate in vitro and in vivo function. So I just want to acknowledge the people that did this work, because I think that's pretty important, since most of the experiments I didn't do. So Stephanie Houtin, a postdoc in the team, really was instrumental in both designing experiments, performing experiments, establishing the collaborations to get these things done. So it's been really impossible. It'd be impossible without her. We have a new thesis student, Alexandra Marinova, who is working on this project and is going to be doing a lot of the SAX experiments. And then, of course, collaborations. So again, from the Centre de Biologie Structurale in Montpellier, Luca Costa, who I met a number of years ago at the ESRF for one of the student science days. So that's been a long-term collaboration for a number of years now. And it's really been instrumental in really advancing our studies. And then, of course, at the ESRF, Mark Tully and Anton Popov, who are more than happy to embark on the studies of LLPS with us. So this has been a great collaboration so far. We're working on NMR with Martin Blackledge. And then for the in vivo work, we have two fantastic collaborators, Phil Wigg and Yvonne Stahl. So thank you very much. And I'll be happy to take any questions.